Hello folks, today we're going to be talking about plant diversity. So without further ado, plants are multicellular organisms that are photosynthetic with cell walls composed of cellulose, and they evolved from aquatic algae. So here is the evolutionary relationship of all of these plants. So they diverge from all the other organisms with the acquisition of chloroplasts. So that's how we get green algae and all of our true plants over here. Once you add in embryos, we get to our true plants. That includes liverworts and everything else. Once we add in stomata and thicker cell walls, so at this node, that's the features that are added in. So everything downstream of that node contains these features. So mosses have stomata and thicker cell walls, but they will lack vascular tissue with xylem and phloem. So ferns have vascular tissue with xylem and phloem. Gymnosperms have seeds, but they do not have flowers. Angiosperms have flowers and fruits, and they're divided into monocots and eudicots, which we're going to talk about more later. So I'm going to ask you about things like, what characteristic do we see arise in angiosperms but not in other plant groups? So we can see here, these are angiosperms. The node right here is fruits and flowers. Everything above it lacks these features which separate them. All right. Which plants have vascular tissue to help with the transport of water and nutrients? So we can see that develops right here at this node. So that is ferns and kin and everything below that. All right. So another thing to understand about this is we are going from very simple organisms to more advanced plants. This is going to be a very important chart. Make sure you understand it, know how to read it frontwards, backwards, understand what we're talking about at each of these stages. If I tell you there is a plant that has stomata but lacks embryos, you will need to be able to tell me that I am talking about a moss, all right? So, purpose of this chapter is to talk about diversity. We'll touch briefly on anatomy and reproductive structures, but really we're gonna harp on that real hard the next couple chapters. So don't worry about that too much right now. Right now we're just worried about the taxonomy and how they're all related to one another. So our non-vascular seedless plants, so liverworts and mosses. So Marcantiophyta, phylum Marcantiophyta, this is a large taxonomic group. These include our liverworts, so non-vascular, seedless, restricted to moist environments because they're non-vascular. They can't transport water very well, so they can't tolerate dry environments too well. They aren't very specialized. They don't have very specialized tissue. They have a thallus, which is undifferentiated vegetative tissue, which is, basic plant functions of life, and the rhizoids, which are kind of like roots, but we don't call them roots because they aren't roots, because they don't do the things that roots do. They are just hold-fast structures. They just hold on. They don't slurp up the good stuff. All right. So these are the two features that we are going to be observing through the microscope. This is the only slide that we are going to be observing for this lab. And so you can bet your bottom dollar that it's going to make its an appearance on the practical. So real quick. All right. So here is our Marcantiophyte in cross section. So this is the thallus, like phallus, but with a T. These are the rhizoids, the root-like structures. These things that look like bubbles, those are still the rhizoids, but if you were harking back to that video of the microtomb where they're preparing slides like this, where they're slicing them into razor thin slices. What you're seeing here is a root. So if you can imagine your thin plane of tissue that's being sliced here, that root is basically curving through, and you're slicing through it like that. And so you get slices of root at an angle, well, rhizoid at an angle that look like bubbles, but that's what you're seeing down here. And so if you see one of these long, almost root-like looking things, those are rhizoids. The main body of this organism is the thallus. You can count on this being on the practical, right? Okay. So here is our picture of it in the PowerPoint. Thallus, main portion, rhizoids down here. Phylum Bryophyta, so our mosses. So pretty straightforward. Most of us can recognize a moss if we see one. Hopefully. That can be a little bit tricky telling them apart from a lichen, which we talked about previously. But fortunately, Inaturalist is pretty good at letting you know whether you're looking at a lichen or a moss. However, there are some species of lichens that have a common name of such and such moss, right? So they are restricted to moist environments because, again, they're non-vascular and seedless. They have a life cycle dominated by the haploid generation. So again, just like the Marcantiophytes, these are very small organisms because they can't transport water and nutrients very well. Non-vascular, remember? So when we first tip-offs when you're seeing on these plants is that it's tiny, okay? You're never gonna see a 12-foot moss, really. I mean, yeah. Of course, there's always exceptions to the rule. I'm thinking Spanish moss, but I think Spanish moss is something weird that technically isn't a true moss, if I remember right. Anyways, so the next phylum that we're talking about is Tracheophyta. So our vascular plants. So those last two groups were phyla. This is a phyla. All of the other groups of plants that we're going to be talking about are classes nested within this phylum. So class Lycopodiopsida. So our club mosses. These are they have vascular tissues. They aren't very well developed. So again, they're still pretty small. When you see these, they'll be a couple of inches tall. They have small, simple leaves. They have spore-producing structures clustered into club-shaped cones called strobili. So this is the strobili right here. Fun little tidbit. These things, the spores are actually somewhat explosive. And people used to go out in the woods and collect these spores and sell them as vegetable sulfur because they would use it. That's what they used in flashpan photography and also powdered the bottoms of babies. So if you're ever out in the North Woods and you find one of these guys at just the right stage of maturity, whip out your lighter and hold it to one of them. And if you're lucky, it'll go up in a puff of smoke. So our Polypodiopsida, so our ferns, pretty straightforward. Most of us know how to recognize fern. There are a few species out there that get a little bit tricky, but generally speaking, you know, we know what a fern looks like, right? So they were the first group of plants to have a well-developed vascular system. This allowed them to move further inland. However, ferns still rely on water for reproduction. You know, they have little sperm that swim through a microfilm of water on the surface of things to get to the ladies. And so they can hack it in very moist environments, areas with a lot of dew and whatnot. But if it's someplace that just hits low humidity for extended periods of time, they can't tolerate it. That's because when they go to reproduce, if they can't, well, after a few generations, they'd kind of be SOL. So sporophyte is composed of fronds, so your leaf-like structure, rhizomes, which is your underground stems, and sori, which are spore-producing structures on the underside of your fronds. So these are our sori here, these little round dark structures on the underside of these fronds. The thumbnail for the project on Inaturalist that you'll be submitting your observations into is a picture of the underside of a fern displaying sori. Here you can see the fiddlehead. At this stage, it would be edible, considered a delicacy, especially certain species. So from here, we're getting into our vascular seed-producing plants, so our gymnosperms and angiosperms. I can put the microscope back under the cover for now. Anyways, so these produce pollen-containing sperm that is transported to the egg, and they can produce in dry environments because they produce these seeds capable of tolerating desiccation. So gymnosperms, that is an old term that isn't really too taxonomically valid anymore. It translates directly to naked seed, so they do not produce fruit. That's what we're talking about when we say gymnosperm. So these are broken down into 4 classes. They have a few characteristics that allow them to survive in drier environments, namely seeds and modified leaves, such as needles or scales. So anytime you see a gymnosperm, it's not going to have traditional leaves like an oak tree, all right? So if you look at this ginkgo, which is in the class Ginkgophyta, it's monophyletic. So the only species, Ginkgo biloba, that's the only one in this entire class. You might have seen it as a health food supplement. Our next class is class Cicadopsida. So these are more palm-like plants. So you see these are its reproductive structures here, not traditional flowers. They aren't producing fruits. It has leaves in the form of basically fronds, like a palm frond. Okay, Canopsida. So these are all of our conifers, so pines, cedars, cypress, that sort of business. They're all Canopsida. The last one is Gymnosperm. So these are our Mormon teas. So subphylum Angiosperm, so that correlates to our Angiosperms. That is the only subphylum nested within the phylum Trachyphyta. Yeah, Trachyphyta. So that means seed in a vessel, angiosperm. So fruits, the good stuff. They produce seeds, more importantly, flowers and fruits. They comprise the majority of plant diversity. They are divided into 2 classes, Liliopsida, so our monocots, and Magnoliopsida, so our eudicots. So 2 cotyledons, which are the first leaf-like structures that you see emerging from a seed, or 1 cotyledon, 1 leaf-like structure. These aren't true leaves. These are basically undifferentiated tissue that was nested inside of the seed, providing sustenance to it. And they will wither as the plant matures and eventually kind of drop off, more or less. So here's a classic example, basically a bean. So 2 cotyledons, 1 on each side. Now our monocots, you'll see 1 cotyledon. So a classic example of this would be corn. If you want to try this out, you can get some dried beans or some bean seeds or corn seeds, put them in a wet paper towel in a Ziploc baggie for a couple of days, and sure enough, they'll sprout. You'll start seeing these structures, and then you can plant them and have yourself a food source because pandemic and whatnot. Anyways, so let's say we find a plant and we haven't caught it right as it's sprouting from a seed. How do we tell whether it's a eudicote or a monocot? Well, you can look at the leaves. So are the veins in the leaves net-like or are they parallel? And we say usually parallel and usually net-like because there are occasionally exceptions to these rules. And so if we just couldn't quite make out these features on the leaves, or if we thought we were being deceived, the next thing we could look at is flower parts. So are they in 4s or 5s, or are they in multiples of 3s? So here we see 5 petals, but up here, I believe these are sepals, not, I don't believe these are stamens. We'll talk more about that in plant reproduction. Sepals, stamens, petals, they all tend to come in similar multiples. So you see these have 5 up here, as well as 5 petals. That's characteristic of a eudicote, whereas this lily has 6 petals, a multiple of 3. So yeah, 6 anthers and 1 stamen. That's what we're seeing there. Sorry. All right. Now, if we really, now also, we might not have seen the flower. Flowers don't appear all year long. Another thing we could do is we could set them in wax and do that thing with the microtomb where we slice them up real thin and make slides like we're going to be looking at over the next couple labs. Now, if those vascular bundles are arranged in a nice, neat ring, that's typical of a eudicote. If those vascular bundles are kind of scattered throughout the middle like seeds in a watermelon, that's typical of a monocot. Now, we could also do this with the roots, and roots are going to be arranged a little bit differently. The vascular bundles in a eudicote are going to be arranged in a little cross-like or T-shaped cylinder. In a monocot, they're going to be arranged in a nice little ring, so opposite of what you see in a eudicote stem. Don't be confused by this. I guarantee we're going to have questions about this, but again, we're going to spend a whole lab on plant tissues, and it's going to bore you all to death, I promise. But that's another day. So your assignment. All right, this is a new one. This is something that I kind of fielded the group for early on. Y'all kind of all agreed this sounded pretty good to you. So I have created a project on Inaturalist. A project is just a place where you organize certain observations. And what I'm asking you to do is go in, and if you don't already have an Inaturalist account, which some of you might have, if so, you're welcome to do it. Use your pre-existing account. If not, go ahead and create one. It's free and easy. My username is Lane Allen, all lowercase, no spaces. That being said, if you can't pick a username that's very close to your real name, or you don't feel comfortable using a username that is similar to your real name, or any of the above, I will need to know who you are, what your username is. So shoot me an email with your Inaturalist username. That way I can give you credit for your submissions. So send me an email with your, I'll add that to this list here in a minute. All right. Sorry, back to what I was doing. All right. So I'll add that in later in the instructions. I'll also create a Word document including instructions for this assignment. And so what I'm asking you to do is create observations of at least 3 different phyla or classes of plants. So, yeah, hold on, let's see if. So go out and try and find something in the class Panopsida. Try and find a fern or a moss or a Marcantiophyte if you can. I promise I will give you bonus points if you can find a real live Marcantiophyte. You'll have to prove to me that you actually took the picture. So write your name on a little post-it and hold it up in the picture next to the Marcantiophyte. That way I know it's really you and not some picture that you found somewhere. Anyways, but yeah. And I will give you bonus points for that because they are really hard to find. I bet none of you are going to find one. Anyways, you're unlikely to find like some club mosses anywhere around here. You might find ferns. You can always go to a plant shop or a nursery or something like, or a park. Quite honestly, this is a great excuse to get up early, take a walk in the park, go for a short little hike somewhere. And I guarantee you should be able to find at least 3 an organism from 3 either phyla or classes. Okay. When I say phyla, you can do Panopsida, and you can do a Liliopsida and a Magnoliopsida. Even though they're all one phylum, they're 3 classes. Sound good? And the thing is that Inaturalist will help you out because it has artificial intelligence, which helps ID plants if you get a good picture of them. That being said, you have to get a picture of characteristic features that are used to classify these organisms. So if you can get a picture of the flower, then it's going to have a much better time telling you whether you're looking at a monocot or a eudicot. It helps get a good picture of the leaves. It's really best if you can get a picture of the leaves and the flowers on all. And you will be contributing to citizen science, contributing to the zeitgeist, anyways. And I encourage you guys to use this just as a tool moving forward. It can be a lot of fun, or it can be a legitimate tool. I mean, if you ever have a tree on your property and you just dying to know what it is, what exactly is this fruit it keeps dropping, pull out Inaturalist, and more often than not, the artificial intelligence can get you pretty close. And then there are other people out here who are going to come along and comment on your stuff and say, I think you're right, or nah, I think it's something else. And that'll help you get some pretty good ideas on stuff that you might not necessarily know much about yourself. For these, you'll need a good picture. You should also include a location and date. Try and be specific about that location. If it's near your house and you don't want to reveal that, there is an option to obscure the location, which I recommend. Okay, that won't affect the way it's graded, and if someone needs to use that data for research, they'll get it anonymously, all that jazz. You can look into that on your own time, but that's one way. You can also set it to private, where basically no one can ever access it, even if they're doing some legitimate stuff. And it just tells the computer where exactly it's set so that it can help it figure out what it's looking at, because it'll filter out certain possibilities based on where your organism was found. Also, at the same time, if you take a picture of a cultivated plant, so something in a nursery or a flower bed that isn't native, that location isn't going to be nearly as helpful for the program. Lastly, be professional, all right? I shouldn't have to tell any of you guys this, but I probably do, all right? If you are not a nice member of the community, the curators can and will delete your stuff, and then I can't grade it, okay? So have professional names, be nice to the other people on here. Yeah, just don't be a jerk, okay? Shouldn't have to tell you guys that. So, real quick before I get into the Inaturalist, let's do a quick review of this. And this is really just because normally, along with this lab, I have a bunch of examples set out so you can, with each of the different clades, so we can say, all of these are club mosses, all of these are this and that, and help you get an idea for what each of these things should look like. And yeah, we don't have live models for this lab, so I do have this little bonus PowerPoint though. So this is our Bryophyta, our mosses, traditional mosses, sphagnum moss. These were actually used as bandages in one of the world wars when there was a cotton shortage, and they found out that these actually, bandages made out of sphagnum moss, believe it or not, actually cause wounds to heal faster and with less complications than cotton. Cotton's pretty nasty. And this moss actually contains a nice, happy, healthy microflora, as well as certain steroids and antibiotics that are naturally occurring in this stuff. Weird, right? So, sphagnum moss, this is phylum Bryophyta. Here is another type of moss. This is Funaria, I believe, off the top of my head. So that's another Bryophyta. These are little sporophytes hanging up at the top, the reproductive structures. So this is our Lycopodiopsida, formerly Lycopodiophyta. Again, restructuring. So if you look close, you can see we have little, these are aspen leaves about 2 inches in diameter. So these shining ground pines are only about 6 inches tall. This is the one that has the exploding spores. This is a ground cedar. This is a little bit smaller still. This is again in the same class, Lycopodiopsida. This is not bearing those strobili just yet. Aha! Hopefully y'all know what class this is. This is Polypodiopsida, our ferns. So I'm going to do stuff like this, where I'm going to give you a picture of a fern and ask you what class it is. Or if I tell you you're looking for a Polypodiopsida, I might say, what sort of plant are you looking for? Liverwort, moss, fern, that sort of business. Lots of different ways I can ask y'all questions about this. So this is a sensitive fern. And the etymology on that one is not very clear. I think it's probably because it dies off very quickly after the first cold temperatures. This is known as the royal fern. There's some mythology that says if you drink the sap from this plant, you'll live forever. When I was there and took this photo, I did not do that because that sounds horrendous. No thanks. All right. So our next group, class Pinopsida, pin as in pines, so needle-like leaves. So this is our balsam fir here. Pretty straightforward. We have little needles. Pine trees. This is a white pine up here. They are both in the class Pinopsida. So our next group, we're looking at this. Monocot or eudicot, how do we tell? Well, affination, it's a little bit tricky. So some of it kind of goes down, some of it kind of goes up. It's not being very nice now, is it? But if we look at these flower parts, those petals come in sets of 3. And so, and also if we look here, these veins don't really branch very much. They do have one branch here and here, but that's about it. They don't do a lot of branching. So this is going to be a monocot. This is in the class Liliopsida. This is the class of monocots. These are more of our grass-like plants. This is a less grassy type. This is our wild iris. You'll find a lot of these growing in wild in Stephenville. Just generational ones that some little old lady planted years ago that outlived her. Anyways, so if you go look out in front of your frat house, there's probably a couple of these half neglected that just refuse to die and keep coming back year after year. Again, you'll notice flower parts in sets of 3. And also, if you'll look over here at those leaf venation, they're parallel, no net-like pattern. This is Clintonia. At this stage, it's a nice, crisp, refreshing salad that tastes of cucumbers. At this stage, those blueberries are violently poisonous, although they're supposed to taste gross. So if you were to accidentally confuse them with blueberries and pop a few in your mouth and chew them up, so long as you're paying at least halfway attention, you should just spit them right back out. Our next group is thimbleberries. So one thing you'll notice here is that it has petals in groups of 5. Can't really count the other flower parts here because they're too numerous, but that's our first tip off. And if we look here, we see net-like venation. So we see a vein one here, here, and here. We see them branching numerous times all over the place. So this is net-like venation. Specifically, this is palmately net-like venation because it has several main veins coming from that central stem. So this is a eudicot, so it belongs to the class Magnoliopsida. This is specifically a thimbleberry. I never got to taste any of those. Apparently they don't produce a lot of berries and the wildlife tends to beat you too. This is wild columbine. We look closely here, we see 1, 2, 3, 4, 5 little flower parts there. So this is another Magnoliopsida. They say you can nip off the nectaries and drink the nectar like, in a honeysuckle. However, to avoid all parts of the stem and leaves and whatnot, because they contain enough prussic acid to induce, and I quote, an unpleasant narcotic effect, whatever the hell that means, right? So this next one we're going to look at is starflower. So if you'll look at this one, you might notice that something funny is going on here. Well, there are 7 petals. Not only that, but there are 7 sepals and 7 stamens here. Okay, I can't really identify which are stamens and sepals in this photo, but that is characteristic of this plant. It's where it gets the genus name Heptaflora, 7 flower. It does have branching venation on its leaves though. Anyways, and that's one of the reasons why flower parts might not always work. You might have to go to one of these other features that we look to. We also have our purple pitcher plant. They're cool because they're carnivorous, just like these Drosera rotundiflora. This was Darwin's favorite plant. He actually said that he cared more for Drosera than he did the origin of all the species. These 2 carnivorous plants have evolved these lifestyles to deal with relatively nutrient poor habitats. And so I've actually, that purple pitcher plant was actually found in a ball of moss floating in a lake. So not a particularly nutrient-rich habitat. Lastly, we have our lowbush blueberry. Delightful. I can't stand domesticated blueberries. I've been spoiled. All right. So a couple examples of our different groups here. Let me stop my share and share. So here we have, I'm gonna opt out and opt back in, make sure we're all on the same page. Hate to make a nice long recording and find out it's all jacked up. So this is my Inaturalist. Let's see. come on. Okay, well, I've got a bunch of stuff going on. I thought I might be able to find that example of Mormon tea for you real easy. Anyways, it looks pretty much just like, oh, here it is. Here, we're getting close. Yeah, right here. Boundary of Phaedra. So, this is our Gnidopsida, all right? So this is our ephedra. This is what it looks like. That's a little bit more zoomed out. It gets a little bit woody down here. All right. So back here, back to my observations. So let me log out real quick. I'm gonna have to move this. So what you'll need to do is pull up Inaturalist. And you will have to log in. You will have to create an account. It's very easy. It takes like 3 minutes, seriously. I'm not joking. Doesn't ask you for any sort of nonsense. Just an email, a username that hasn't been used before, and a password, and to confirm that password. So pretty straightforward. Here I have created a example account just to show you what we're doing here. Don't say. Log in. Come on. Cut to it. Okay. So we go to your observations as this test student. And this is what it's going to look like for you when you first join on. Nothing going on. It'll pop up at your dashboard. So it says, hey, why don't you post some stuff? That way people can talk about it. And gives you a little bit of a place to start from. You can update your profile and all of that business, but for the important stuff here, you've got to be able to make some observations. So what you would do is you'd click upload. And so then it's going to pull up this screen. So then you're going to choose some files. And so let's see, I'm going to go over here. I'm going to choose this nice bryophyte that I photographed earlier, and this Virginia creeper. Click OK, and it uploads them. And because they're in the same area, I'm going to click select all and edit their location. Then I have to scroll in. This is a little bit tedious. However, if you're using the mobile version and you want to just use your iPhone, it's real easy to just snap a picture and it automatically uploads the time and location. Anyways. And so for the purpose of what we're doing here, that's good enough for me. Maybe a little bit smaller. Anyways, because this is close to my home, I'm going to leave it as obscured. You can do private if you want, but you need to set the location because that'll help the artificial intelligence decide what exactly this picture is of. So update our observations, and we see now both of our observations, which have been highlighted in green here, right around the border, have location tagged here. So now we need to say what we think they are. Now, your IDs at this point don't really have to be correct. What I'm looking for is for you to get the class, or if it's a moss or a liverwort, the phylum correct. Okay, so on this one, if you just click the species name and give it a moment for it to mull it over, so the artificial intelligence is taking in the location and the time, and this photo is taking sweet, sweet time. First, I guess I've got a lot running. There we go. So when it says we're pretty sure it's in the genus this, well, they tend to be right. And so then you see, then they're saying, now what we think it's probably one of these things. And if you look at their suggestions, a couple of them are this Climasium, a couple of them have a different genus. So they're less related to these mosses. If we get down here to this fragrant flat sedge, that is not a moss. So anyways, let's go ahead and click this one. Actually, let's click view. And so what it's going to do is it's going to open up a new tab, and now you can compare it. All right. So these are existing pictures of mosses in that genus. You can check out similar taxa. Make sure you aren't making a mistake. See if one of these look just like what you've got. Honestly, this kind of looks like what I've got, but I'm not being that picky right now. I just don't know that well. Quite frankly, again, for this particular lab, it's not that important. But if we go over here to taxonomy, look at this. So here's our genus, Climasium. Here are the species. And way up here, you can see that they belong to the phylum Bryophyta. All right. And so this would take care of one of your phylums or classes. Do not repeat phylums or classes. So don't give me 3 mosses. Don't give me 3 monocots. Give me 3 unique classes or phylums that we discussed in that PowerPoint. Okay, sound good? All right, so back to this. So also, the other thing too is, let's view those suggestions again. And let's open up this. And let's view that. And let's check out the taxonomy there. Well, first of all, you can see this is a great big old plant. It has real leaves, and that is not what we're looking at. What's happening here is it was picking up the grasses on the margins of that picture, and it's saying, I think there was a sedge in that photo too. That wasn't what you're talking about, right? Anyways, and that's why it makes these sort of mistakes. Anyways, but if you go to taxonomy and go way up here, you can see that this particular plant would belong to the phylum Tracheophyta, and specifically to the class Liliopsida, because it is a grass-like plant. It has parallel venation in those leaves. Anyways, so I know that this is some sort of moss. So for the time being, I'm just going to call it whatever Climasium. And this one, I know it's Virginia creeper. So I'm just going to go ahead and start searching for it. Virginia creeper. Now, again, if you aren't sure what class this belongs to, go to the information on this particular plant and go to the taxonomy section and look up here, phylum Tracheophyta. Pretty cool, right? And it tells you all this stuff, like it's in the grape family, it's in the order Vitales, grapes and allies. And this is important because plants that are similar will often have similar compounds and similar properties. There's a lot of uses to that. Anyways, like, for example, if any of you have ever heard about the first time people started eating tomatoes, the story goes, well, for a long time people thought tomatoes were poisonous because they're in the nightshade family. Fun fact, potatoes, also in the nightshade family, produce a bright red fruit that is deadly poisonous. They produce these tropane alkaloids that make you feel kind of funny and then kill you. Anyways, and they're really toxic, and it's in basically all parts of these plants that we don't eat. Anyways, everyone thought tomatoes were deadly poisonous. Anyways, one day this guy finds out that they aren't somehow. And he makes a post in the town square and says, I'm gonna eat a basket full of tomatoes. Come and watch it, buy a ticket. And so everyone comes and buys a ticket because, you know, they didn't have Netflix or, you know, quality entertainment and whatnot. And so they're like, hell, let's watch this guy poison himself to death with tomatoes. And so they all show up and they just stare all slack-jawed as this guy plows through a basket full of tomatoes. And then they sit there expecting him to keel over any minute, and he doesn't. He just sits there looking at him. And the story goes that he made a killing selling tomatoes to everyone. I don't know how true exactly that is, but yeah, that's the reason why, yeah, knowing how plants are related is important, because it helps you know which ones are poisonous and which ones produce good foods and that sort of business. All right, so our Virginia creeper, I know what it is, so I'm just going to click that. And we've come over here, we can see through the taxonomy that it is in the phylum Tracheophyta, and specifically it is a dicot, which is sometimes used interchangeably with eudicot. Again, just old terms, eudicot. Anyways, but it belongs to the class Magnoliopsida. Okay, so for these two observations, I have phylum Bryophyta and class Magnoliopsida. If you give, for this test student, they would need to send me a picture of a grass or a fern or something, or a pine tree or something, a cedar. You know, any of those would be valid submissions. So yeah, take this as an excuse to get up in the morning, go for a walk through the park, take a couple of pictures of plants and start uploading them to Inaturalist and check the text. Make sure you see what their IDs they suggest, make sure they make sense to you, make sure the pictures match up, they look alike. And I'll come in and check them, I'll let them know if you got them wrong, and then you can go back and fix them. If that is the case, shouldn't be too much of that. Anyways, give me 3 classes or phyla, make sure they're all unique, and that'll be full credit for this assignment. Later on, I'm gonna have another assignment where I'm gonna have you go out, take a picture of some sort of Angiosperm, a flowering plant, and then I'm gonna have you in this notes section describe things like the venation, the leaf arrangement, that sort of business. Anyways, for now, that's all. So until next time, it's been wonderful. Don't hesitate to send me any questions you have. Please email me. Do not use Canvas. I can't guarantee that I'm going to see your Canvas messages as soon as I'll see emails. All right. Oh yeah, and the last thing, submit my 2 observations. Oh, sorry. Was getting way ahead of myself there. I almost forgot one last bit, okay? How to join the project. So you come up here to community, go to projects. So sorry, I hope you all didn't just jump off when I started sound like we were done there. So search TSU. That should be all you have to search. And mine's way down here at the bottom because I'm the new guy. And you click on TSU Biology 1407 Plant Labs. And so go up here. Here's a picture of Timberlake Ranch. Here's a thumbnail of a fern exhibiting sori on the underside of a frond. Anyways, and you need to click join. So then it brings up this next little screen saying, are you sure? And then here are the rules for it. So, my stuff isn't going to show up here. It can't be an animal or fungi. So if you upload those, it isn't going to automatically be uploaded to this. I don't know how to curate these better because I don't know how to make it where it's only certain things, but I'll curate them. Also, I don't think y'all are going to be terribly active in between the time that we get started on this and the time I finish grading them all. Anyways, but it must have a good photo. If your photo doesn't provide enough evidence that I can say it's this class or that class, I'm going to tell you to try again. All right? You do not need to receive updates from this project. You can if you want to. And you click yes, I want to join. And bam, now it says 2 members of this project. So I need you to create an Inaturalist account, send me an email with your username. That way I know which student gets what credit. Go out, make 3 observations of 3 different classes or phylums. Should be a reasonable task. And then make sure you have this done by the time all practical 2 tasks are done. And that'll count as one of your assignment grades. Your 3rd assignment will be in the practical 3 section, so we'll worry about that then. Anyways, now I've finished it all. I've made my piece, and we can all part ways. So until next time, bye.